Hello, I'm Denethi from the Gopal Lab, Department of Pathology at Yale School of Medicine. Today I'll be talking about how ataxin-2 polyglutamine expansions aberrantly sequester TDP-43, impair anterograde transport of neuronal RNP granules along the axon, and suppress translation. ALS is a debilitating and fatal disease characterized by progressive degeneration of motor neurons, nerve cells in the brain and spinal cord that control muscle movement. Almost all patients with ALS have pathological aggregates of TDP-43, an RNA binding protein with multiple roles in RNA stability and processing. Mutations in TDP-43 cause familial and sporadic ALS, but it's unclear how defects in RNA biology cause disease. In healthy cells, TDP-43 is nuclear, but in unhealthy cells, TDP-43 gets mislocalized to the cytoplasm. We and others have shown that TDP-43 is a component of neuronal RNA transport granules, and these are composed of RNA and RNA binding proteins. In previous results, we have shown that ALS-linked mutations disrupt TDP-43 transport and dynamic properties of neuronal RNP granules. So most ALS patients actually don't have TDP-43 mutations, but still they form TDP-43 aggregates. So we asked, what are other additional mechanisms that regulate biophysical and functional properties of neuronal RNP granules? And what makes them go from these soluble RNA-protein interactions in physiological conditions to less dynamic aggregates in pathological conditions? This is where ataxin-2 comes in. So ataxin-2 is a genetic modifier of TDP-43 toxicity. In a Drosophila model, TDP-43 expression leads to degeneration of the retina. The expression of TDP-43 with ataxin-2 leads to a much worsened phenotype. And when you delete one copy of ataxin-2, it suppresses this TDP-43 toxicity. So ataxin-2 is an RNA binding protein which contains a trinucleotide repeat encoding polyglutamine. This repeat length is normally 22 to 23 glutamines, and long expansions of ataxin-2 polyglutamine tract greater than usually 34 polyQ cause spinocerebellar ataxia type 2. Any intermediate length polyglutamine expansions between 27 to 33 glutamines increase the risk for ALS. So we wanted to investigate the dynamic interactions between TDP-43 and ataxin-2 in primary neurons. Our hypothesis is that ataxin-2 polyQ expansions aberrantly sequester TDP-43, disrupting RNP granule transport and function. Subsets of TDP-43 and ataxin-2 granules colocalize with known neuronal RNP transport granule markers such as PABPC, FMRP, and G3BP. TDP-43 and ataxin-2 colocalization with each other is about 25%, and this colocalization occurs more in the proximal axon compared to the mid-axon. We also see that ataxin-2 is present more proximally in the axon, while TDP-43 is distributed throughout. We then examined the transport of RNA granules containing TDP-43 or ataxin-2. We expressed fluorescently tagged wild-type TDP-43 or ataxin-2 in rodent primary cortical neurons and quantified their motility along the axon. In agreement with previously published work, we find that TDP-43 positive granules are often oscillatory or show long-range motility of greater than 10 microns net distance. In contrast, RNA granules containing ataxin-2 are predominantly stationary. The cumulative distance for ataxin-2 is much lower than that of TDP-43. We also found that RNA granules positive for both TDP-43 and ataxin-2 are stationary. Next, we asked how longer polyQ expansions in ataxin-2 affect the TDP-43 motility. So here we have co-expressed EGFP-tagged wild-type TDP-43 with either a control plasmid or with M-scarlet ataxin-2 of different polyQ lengths. Changing the polyQ length didn't really substantially affect the fraction of stationary, motile, or oscillatory granules, but what we did observe was that the longer polyQ, so ataxin-2 Q30 or Q39, co-localize more with TDP-43 granules. This suggests that polyQ expansions sequester TDP-43 in double positive granules. More TDP-43 is entrapped within the longer polyQ length ataxin-2 granules. In support of that, in neurons expressing ataxin-2 Q30 and Q39, the TDP-43 anterograde run lengths are decreased. You can see the decrease here. This is a histogram showing the position of TDP-43 granules along the axon, with the longer polyQ length shifting the position of TDP-43 granules proximally. So these granules don't travel as well to the distal parts of the axon and are trapped proximally. We then did FRAP experiments to test whether ataxin-2 polyQ expansions impair TDP-43 dynamics. The FRAP recovery of TDP-43 in RNP granules expressing ataxin-2 Q30 or Q39 is reduced, indicating reduced dynamic exchange. So the intramolecular mobility of TDP-43 is decreased. Finally, we asked whether RNP granule function is affected by the longer polyQ lengths. So I have carried out puromycinylation assays which show reduced translation in neurons expressing ataxin-2 with the longer polyQ lengths. This is also denoted by the fewer puropuncta being present as the polyQ length increases. In conclusion, these results indicate that ataxin-2 polyQ expansions aberrantly sequester TDP-43 and affect the spatial localization, transport, and dynamics of TDP-43. Preliminary data suggests that translation is inhibited in neurons expressing ataxin-2 polyQ expansions. This may have detrimental effects on localization and translation of transcripts that are critical for neuronal axon integrity. With that, I would like to thank my PI, Dr. Gopal, Junji, Stanali, and also the Yale CNNR Core, NIH, and ALS Association. Thank you.